Everyone? Do we have everyone? From the floor. Okay, we are gonna go ahead and get started. Just want to make one quick announcement before I pass it over to the next panel. We so much value the feedback of everyone who's participated in the last couple days. So you will be receiving a survey via email. It's also live in the event app if you downloaded this event app. Please, please, please share your feedback, share your thoughts about the event, about the sessions. We do all of this for you guys, and so we really want to know how things were this year, what we can do to make it better, what topics you'd like to hear more of. So we really encourage you to provide that feedback either directly to CPAF staff or through the survey. So with that, I'm going to go ahead and pass it over to our next panel.
Thanks a lot. Microphone's on. Thank you very much for joining this session. This is going to be a slightly more technical session than perhaps some of the previous sessions, but it's a fascinating topic, and I hope I can, if I need to convince you the same. I'm Tomas Salmonson. The picture is 10 years old, as you can say, but it's a little bit more gray, but sorry for that. And I note I'm not the first one using that trick. We have an excellent panel today. Marie, Smithy, Rob, Angela, and Ralf, and I'll ask them to introduce themselves when they make their remarks later on. But let me try to frame the problem for you. And I'm not a statistician, so I do it in my layman's term, just so we understand why we think this is an important issue and what we hope to achieve today, addressing a couple of questions. I'm a dinosaur, and I'm also very traditional, so I love the randomized controlled trial. And Peter understands why I love the randomized controlled trials, because it sort of isolates the drug effect. We have inclusion-exclusion criteria to ensure that we have a population that we understand. We have a randomization to make sure that the two groups hopefully are quite similar. We blind the study often, so there's no difference when it comes to placebo effect. And all this, hope I'm not oversimplifying things, but usually we can then interpret any difference between groups as being related to a drug, right? That's good, easy for us, relatively speaking. We've also heard during this meeting, and we know it very well, that science delivers fantastic new drugs. New drugs for orphan diseases, new drugs that has an impact on disease modification that takes a long time to see the true value of, but are really changing the course of a disease and have a huge potential. But these new drugs, they present the problem that sometimes randomized controlled trials are not feasible. And the second best option is often to do a single-arm trial of the drug and compare to an external control data that's coming from data generated in clinical practice, or as some people want to say, real-world data. And that is where some of the methodological difficulties may arise. Because if you think about a placebo, if you think about the kind of well-defined patient population that goes into a clinical trial, the placebo effect, the trial effect, I bet that if you take a placebo group in a randomized controlled trial and just crudely compare it to an external control, the placebo group always wins. But both untreated with something active, the placebo group always wins. And for that same reason, if you take a single-arm trial with a drug, hopefully active, it's always going to win, most often at least, compared to an external control group. So how can we know that what we're looking at is actually a drug effect and not a consequence of greater placebo effect or a selection of patients with better prognostic factors or anything else. And this is not a new problem. This is a well-recognized problem. There are loads of papers published. There's many regulatory guidelines that we will hear about later on. But in my mind, it still presents a major problem, and it's a barrier to fully use the external control type of situation. So the question for you and for us in the end here is, do you agree that we, despite recognizing this issue of comparing to external control, that we have a challenge in implementing it in drug development? And if so, you agree that we perhaps can move forward, and perhaps, coming back to Smithy at the end, is there a role for CPATH in developing part solution to this? Because we're probably not going to solve this with one type of tool. We probably need a number of tools to progress this and facilitate interactions within the sponsors, discussions initially, but then also between sponsors and regulators. So that's framing it. Peter, was that clear? Thank you very much. I paid him. So we're going to have two slightly longer presentations to framing this problem in a better way than I did, in a more sophisticated way than I did. I'm a brutal Swede. And that's Angela and Rob. And then I'm going to ask the two regulators and Smithy to make some introductory remarks, and then we have a Q&A session. So I hope that works for you. So, Angela, you're first up. Please introduce yourself, and we're looking forward to your presentation.
Well, thank you so much for setting up the discussion so nicely that we are about to engage in. I'm an epidemiologist by training, and I have 15 or so years of experience in observational and clinical research, starting in academia, and then within the last 8 years within pharma, focusing on rare disease. And currently, I have responsibility for real-world data generation in neuromuscular diseases, with a particular focus in spinal muscular atrophy and in ALS. And much of my time and focus really goes in towards ensuring that we meet standards that enable the end use of these real-world data to meet the various needs of stakeholders, including regulators. And so this includes collaborating with registries to meet post-marketing safety and efficacy requirements. It includes label updates based on real-world evidence in populations outside of the pivotal studies, and it extends to use of external control arms to really contextualize the findings of single-arm studies that Tomas pointed to. And our focus today, as he so nicely pinpointed, is really the use of real-world data to supplement, or even in some contexts, replace control arms in randomized control studies to make efficacy determinations. And so to set the stage for our discussion over the next few minutes, I'll provide a perspective from industry on key considerations for using external control arms based on real-world data in pivotal studies. And Rob will discuss some of the more detailed methodologic issues arising when such approaches are used. We'll also, if I can advance, let's see. Yes. We'll also explore the role of a potential toolkit that was mentioned in standardizing the planning and proposals of use of external control arms, which I'll call ECAs, and outline important design considerations, at least from the industry perspective, for a tool aimed at facilitating better alignment between sponsors and regulators. And before I begin, I, of course, want to note that I only represent one industry viewpoint, and I'm sure if you had other colleagues out today, they may have different perspectives. So real-world data has long been used in regulatory decision-making and routinely included in submission packages from sponsors. For instance, epidemiologic analyses provide important information about prevalence and incidence of disease, disease state, and natural history. And real-world data have also been used for post-marketing drug safety monitoring, as well as included in and help support label changes. And increasingly, companies are really exploring a more ambitious application. So external control arms based on real-world data sources to support or make efficacy determinations, and particularly when doing so in contexts where it can lead to efficiencies in the drug development process. But the path for this is not always clear, especially when considering the rigor needed if external control arms are being considered to replace traditional randomized control arms. There are multiple relevant guidance documents from FDA, EMA, and other regulators. They provide a high-level framework and broad considerations for covering important topics for conducting real-world evidence studies. This includes use of specific data sources like electronic health records or registries, as well as the design and conduct of externally controlled trials. And the FDA guidance specific to the use of external control arms provides rather comprehensive, high-level overviews of key study design and analysis components that need to be considered. But from my perspective, the guidances are high-level and certainly not binding, and they emphasize that ultimately sustainability or suitability is determined on a case-by-case basis. And additionally, there's not clear guidance on the core mandatory requirements, as well as which elements could be adapted to fit the scientific and practical context of a specific disease area, right? Not all standards applicable to clinical trials can be directly applied. And the context, of course, also varies by therapeutic area and in specific circumstances. And as a result of this, industry is often left uncertain about whether a proposed approach will be acceptable to regulators. So currently, external control arms are feasible in narrow drug development specific areas, and I've included a few on these slides. This would include rare diseases where randomized control trials are infeasible or unethical, certain oncology indications, particularly in high unmet need populations, as well as their use in augmentation of single-arm trials, with the addition of externally collected natural history data. There are other emerging uses, including in pediatrics, gene and cell therapies, post-approval label expansions, and so forth. But even in the areas where external control arms are being used, challenges do remain. And I've listed a few here, which we'll explore a bit more in a moment. But I would argue that a toolkit could potentially help sponsors evaluate feasibility, identify gaps, and plan mitigation strategies upfront, and really prepare for meaningful discussions with regulators. So from my perspective, there are several key challenges in this space for industry. So I mentioned regulatory uncertainty. So I'll just spend a moment defining what I mean by that and how it arises. So for me, regulatory uncertainty arises in part because standards for data quality, methodology, and evidentiary weight are still evolving, right? And their acceptance does remain case by case. And without early regulatory alignment, sponsors also face the risk of late-stage rejection after significant investment in data collection and analysis has already occurred, which can provoke, some anxiety as well as uncertainty. Additionally, there are challenges in identifying and selecting high-quality fit-for-purpose data for external control arms. Real-world data sources can vary widely in accuracy, completeness, and relevance, and there is ongoing uncertainty about which data sets, regulators would deem as acceptable. And an example is given here about data considerations and standards, such as the need to harmonize endpoints, since outcomes captured in real-world data often differ from those in clinical trials. And as I spoke about a minute ago, there are limited precedents for the use of external control arms in efficacy determinations and in pivotal trials. So many sponsors use external control arms in parallel with traditional trial evidence rather than relying solely on them to de-risk their regulatory submissions. Whoops, I'm going to go back just for a moment. Ooh. I don't know if I can get help. Thank you very much. But I would like to, before I close, talk a little bit more about how a toolkit could potentially enable or overcome some of these challenges or provide confidence, right, from an industry perspective in terms of, relying on the use of external control arms. So how could a toolkit be used? Really to support pre-submission self-evaluation by sponsors of key scientific and regulatory requirements. And I really do think that these tools, if we could go back a slide, would be most helpful if they could help us understand whether an external control arm approach would likely be acceptable from a regulator's perspective really early in the process. For instance, the tool could help clarify regulatory expectations by, for instance, summarizing FDA guidance, providing case examples, as well as acceptable methods. And this self-evaluation process helps with broader planning as well within the industry context to make sure we have the right resources, the right timelines, and as well as the right expertise at the table. It could also provide shared language for regulatory discussions, helping sponsors prepare clear evidence-based proposals for productive FDA meetings. It could help encourage consistent, high-quality proposals and guide methodologic planning, including, perhaps clear outlines of what a protocol would look like, because I think the protocols themselves for studies that are based on or incorporate external control arms look very different than what many folks in industry are used to within the context of a traditional clinical trial. Prompt structured consideration of bias, mitigation strategies and sensitivity analyses, and a process to ensure scientific rigor and transparency as well in the conduct and reporting could be furthered by having a toolkit that can really lay out very clearly what's needed from a sponsor perspective. So in essence, what I'm suggesting is that a toolkit could act as both a decision-making guide and as a communication aid, making regulatory interactions more predictable and more efficient. So here, just in brief summary, I do believe that industry, wants to be able to use very thoughtfully external control arms in pivotal studies, but clarity, consistency, and confidence in doing so are absolutely essential. And embedding considerations for key requirements from regulators' perspectives on scientific rigor and acceptability into a toolkit could really help enable sponsors to make better, more informed assessments of the feasibility and appropriateness of external control arms really very early in the development process. And while use cases might be somewhat narrow and context specifically, particularly as we think about their replacement for randomized control arms, this approach can help ensure that when ECAs are considered in pivotal trials, right, they are applied thoughtfully and with rigor, with the intention of benefiting patients, regulators, and sponsors alike. So with that, I will turn it over to Rob.
Yeah, thanks, Annie.
Wonderful.
Musical chairs.
Thank you.
Thank you. Thanks, Angela. Thanks, Thomas. Rob Hemmings. I'm part of the CPATH board in Europe. Long time ago, I was trained as a medical statistician, and I had about 20 years in the European regulatory system for medicines. We all know, we all understand that industry run clinical development programs to learn about the effects of their treatment. Regulators, excuse me, bad start. Regulators need to make decisions based on the effects of those treatment. Has efficacy been established? What's the magnitude of efficacy? What's the safety profile? And eventually to make a benefit risk decision. We all probably also know that randomized controlled trials have some limitations, perhaps the external validity of the experiment, perhaps the choice of endpoint. But as Thomas touched on, the well-controlled conditions of a clinical trial is great for understanding the magnitude of the change in a patient's condition that's due to treatment rather than due to other factors, i.e. it's a wonderful experiment to try and estimate the treatment effects. When we randomize, we compare outcomes in the same patient population under the same experimental conditions, including when and how outcomes are assessed. But some studies, of course, are at the boundary of equipoise. If we go into a large phase 3 study, we've already got some idea that the drug will be beneficial. And so there's interest in reducing the size of control arms or replacing them entirely with external control groups, in particular when there's a risk of exposing patients to a suboptimal control arm. And we can think about two scenarios. Firstly, where no RCT is feasible, either because of clinical or ethical issues, or because the sample size you could recruit is not sufficient. And then we have to do something different, right? We're forced to do something different. The adequately controlled RCT is just not an option. Perhaps more interesting to discuss is situations where the RCT is feasible, an adequately powered RCT is feasible, but where the sponsor thinks there's adequate justification for the use of an external control arm. And that's really what we'll talk about today. Indeed, this seems to be the trickiest part of using real-world data, real-world evidence in drug development and as part of regulatory decision-making. In my 6 years since leaving the regulatory system, and my experience is predominantly watching EMA and watching the EU, but in my 6 years, there seems to be a disconnect between policy statements around real-world data, real-world evidence, and the willingness of regulators to accept specific proposals for its use in the generation of substantial evidence of efficacy. Many, many ideas that come across my desk as a consultant that eventually don't get traction. And do not think for 1 minute that that's a criticism of the regulators. What that represents is a misunderstanding in industry about how difficult this is. Planning this type of experiment is an order of magnitude, at least one order of magnitude, more complicated than planning an RCT. So we're in the bottom left-hand corner. We're talking about real-world evidence to supplement or to replace control arms. And as you heard in Thomas's introduction, the fact that we're comparing patients between different data sources, a clinical trial on one hand and a real-world evidence source or real-world data source on the other hand, and we're at risk of comparing different patients, a different type of patient goes into the study than is in the natural history or in the registry or in the medical records or wherever you might be getting your real-world data from, risks the introduction of bias, so that when we estimate our treatment effects, those estimates are unreliable. They don't correctly quantify the magnitude of the treatment effect, which can, so that the estimates can be unreliable for regulatory decision-making, and patients risk being misinformed about the effects of treatment. And as Angela touched on, there's multiple regulatory guidelines in the area of real-world data. We'll probably talk mostly today about the FDA guideline from
2023, early 2023, on external control arms, which nicely outlines the risk of bias in this type of comparison. And I should have done this slide as a build. So we're going to go from top to bottom. I'm going to start at the top. So we have this risk of bias. And the sponsor needs to think, what assumptions am I making? What limitations are there in the data? So what potential biases and confounders might be introduced when I'm trying to estimate treatment effects using an external control arm? What risks of bias need to be addressed? And here, the regulatory guidance documents give a nice outline to sponsors of the type of things they should be considering. Having identified the risks, the sponsor then needs to think, what shall I do about it? And so they think about sensitivity analysis, they think about the investigation of potential bias. The FDA guideline, again, asks for sponsors to provide justification for the methods that they select, describing their strengths and limitations, identifying and managing sources of bias, making assumptions explicit, and examining those through qualitative and quantitative bias analyses. But the FDA guideline can't be a textbook. It can't be a textbook on statistical epidemiology. And it's left to the sponsor to think through what data presentations and analyses do I need to do for my particular case. Mm-hmm. And not all sponsors are competent to do that. Not all sponsors have the in-house competence or can access the expertise to be able to do that well. And then, as Marie touched on, having done that, we have to think through what tolerance will there eventually be for risk of bias in the regulatory decision-making. Is the mindset of the regulator here, yes, the company has done the best they can do, or is the mindset of the regulator here, is this evidence going to be as reliable as an RCT? And if that mindset is the latter, which again, I get, if the mindset is the latter, this is a really high bar to clear, okay? And some assumptions we are forced to make in this type of comparison are essentially untestable. We can't know whether the assumption is valid or not, and there will always be an uncertainty of a risk of bias, perhaps even a major risk of bias, that can influence this type of comparison. And so back to Tomas and the discussion today, which is beyond the regulatory guidance, is there a drug development tool? Is there a toolkit which would help sponsors as they prepare for those regulatory interactions to discuss specific proposals? Excuse me. And now I've missed a slide.
Oh, you missed a slide.
One more word. I was so keen to sit down before I coughed. One more word. Of course, we're here at the CPATH conference, and part of the solution here, or part of making progress here, might be that we still don't quite know which data presentations, which methods, which levels of tolerance can be most useful in a given situation. And what you would need to develop that and to investigate that is access to data. And of course, that's what CPATH has. And if we have an area, for example, where we've got clinical trial data and we've got real world data, we can start to do investigations and perhaps try and optimize, not just identifying what sources of bias are important, but the types of data presentations and analyses that best illustrate the risk of bias or quantify its potential magnitude. Again, trying to inform those regulatory discussions. I'll stop this time.
Thanks, Rob. But that's really good, and I'm a very proud board member of CPATH, and of course, when it comes to what Rob just talked about, a toolkit that may involve disease models or using a large amount of data from real world as well as randomized controlled trials, I think that speaks very nicely to potential role of CPATH. So that's why we're having this conversation. So thanks, Rob. So I'll now turn to our two regulators. And Marie, the FDA paper from 2023, right, was mentioned. So I go to you first. And if you have any opening remarks.
Is the mic on?
Yes.
Yeah, okay. Yeah, so my name is Mari Bradley. I am a senior advisor for real world evidence in the Office of Medical Policy in CEDAR, currently the CEDAR lead for real world evidence. And more recently, I've been working part-time in the Office of the Commissioner as an advisor on real world evidence for them as well. So I'm very happy to be here today. You know, I think I'm just going to focus on, you know, the challenges and the concerns have been outlined and just some of the activities that FDA are currently engaged in to try and, you know, encourage robust design of externally controlled trials. And so, you know, we mentioned the guidance, we received an overwhelming amount of responses to that guidance from the public, and we're working on integrating those into more into revised guidance, which may, in a sense, address some of the gaps that, you know, Angela and Rob were alluding to. I would also like to mention that, you know, consulting with the FDA early and often is really, really important, and especially in these areas where there are several technical challenges and it's a new area, and so we're very open to, you know, giving meetings, Type C meetings, where, you know, as often as we can, and really we have a multidisciplinary team, statisticians, epidemiologists, clinical reviewers, you know, attending those meetings and providing even written responses to some of the questions. So that's certainly an option. We also have an advanced and real-world evidence program, and so sponsors who are potentially considering using an externally controlled trial can submit through that program to get more focused attention on their submission before it goes through traditional channels. I do want to highlight again that, you know, yes, sometimes the externally controlled trial using real world data is appropriate because the trial isn't feasible or because, you know, but it needs to be in a situation where the natural history of the disease is well known and where large effect sizes are expected. It's not just a one size fits all. I think it's really important to emphasize, and externally controlled trials are held to the same regulatory standards in terms of generating, you know, substantial evidence of effectiveness from an adequate and well-controlled study. And so I guess we try and put out guidance to help, you know, people with, you know, submitting something that's going to meet that that bar. And so, you know, there are several study design considerations within the guidance. It's been mentioned already. Comparability of the two arms is this is one of the probably the major barriers we see in submissions where, you know, the groups are just not comparable with respect to demographics, the socioeconomic status, geographic region, disease attributes, prognostic factors, comorbidities, current and prior treatments, even imbalances in treatment between the RCT arm and the, you know, the external arm from real-world data. You know, in a trial, people tend to be more adherent, right? In clinical practice, it's not always like that. They may use different doses of treatment. They may use it for longer durations. Also, you know, in terms of outcome assessments in a trial, they're always much more protocolized and standardized, potentially using imaging that's just not done in clinical practice. And often outcome assessment is done at different frequencies in a trial compared to real-world data. And so all these challenges, and along with, you know, concerns about appropriately, you know, indicating, selecting an index date, which, you know, the index date's important for multiple reasons, which I won't go into in these preliminary comments, but really trying to avoid a mortal time bias. And that's a key, I would say one of the key challenges that we see with a lot of these submissions is misspecification of the index date. And also, so that's our guidance, right? And, you know, we're working on getting this finalized guidance out and responding to all the very good comments that we've received. But in addition to that, we also fund a large portfolio of research. And so we funded J&J to look at measurement error when using externally controlled external controls from real-world data in multiple myeloma trials. And they developed like a statistical approach that can help to provide context to the study findings in the presence of measurement error, like, you know, when outcome is misclassified, things like that. We also funded Genentech UNC and Duke to look at sort of methods to advance the use of hybrid externally controlled trials where you have a combination of trial and real-world data in the control arm, which is even more complex and even more complex concerns. So, you know, we're investing heavily in that research. And then recently we funded the University of North Carolina to look at a project that is concerned with approaches for appropriately selecting the index date that I mentioned and how these different approaches to selecting the index date can affect the study findings in different oncology settings. So we have quite a wide portfolio, just really to emphasize that we understand the challenges. You know, we're working on the guidance, we're working on this research, and as I mentioned, we're very open to early and frequent discussions with anyone who's considering these types of designs.
Brilliant. And that, again, it very nicely illustrates the point made by Rob previously. I think regulators probably understand this far better than many sponsors. But Ralf, something from the EMA perspective on this one as well?
Thanks, Thomas and Rob and Angela, and Marie, thanks. This is really a hot topic, but I personally feel the heat here. It's super hot up here. I'm not sure. Just me, if not, maybe be made a little bit more comfortable, and I have to also go to
the position down there was quite cold, actually. Yeah.
As a disclaimer, I'm Ralf with the European Medicines Agency since a longer while in different roles. I now lead the regulatory science and academia workstream, and you need in your mind, please, to expand it to doing regulatory science research on topics that cut across the agency's activities with not-for-profit organizations, with academia and others. One of the reasons I'm here. I don't speak for the whole of the agency or even the European network, etc., but that just needs to be said because we have thought about what we can say here. And let me start that I primarily want to focus on the CPATH-related questions. So is there something to be done? And of course, I think that that's relatively easy. We would like to explore that area. So what could be done and then do it together. But let's look at this from the research perspective, the drug development tool research perspective. In that sense, we need to investigate a couple of these scientific aspects that have been mentioned. And to do so, we have, similar to the colleagues at the FDA with whom we also collaborated, the ORC colleagues, or when the ORC existed, published regulatory science research needs. And I'm not sure if you've seen them, but they exist now in the recently updated version. And they come out of an exercise where we collect issues that people see for the first time and second time and third time. It becomes probably a research topic that we want to formulate. And this includes, for example, research into how natural history studies can be used as comparative groups or groups for comparison. And at the moment, we are still working also with the Methodology Working Party and others to have some more research needs. So this is an acknowledgement of that topic area. In previous public partnerships like EU Pearl, we already heard about the Bayesian time machine, you know, secular trends, etc. So I think there's a huge interest in the science of it. And I think this is also an important aspect, and two more remarks on that, since we need to also bring the relevant researchers together. And I'm thinking of bringing, let's say, ISPOR and FUSE together, or the other organizations where you have pharmacoepidemiologists and you have biostatisticians, and some are working together, but when there was a question about what further methods are needed, some of my colleagues says, yeah, but we do have methods for quantifying bias and so on in pharmacoepidemiology. And I think there is one aspect in making then sure that the topic is explored with all the relevant communities. The second but last comment is that we regulators may not see all the cases. There is a lot of decision-making in companies, and then an argumentation is built on top of the decision. And we then see that big document and the many considerations, but we haven't really been fully, or we are not easily able to embrace the whole challenges from the outset, based on which then companies take a decision and then make a make an argument that the RCT is unfeasible, etc. So, but I think that is therefore why, last point, perhaps it's important to think of a workshop or some way of scoping topics and getting them together to abstract the issues that you have seen and find that level where pre-competitive research activities are helpful. We have mentioned a couple of these issues already. You also iterated this. Our recent guideline on single-arm trials goes into itemizing biases and what could be done about it. And so there is a big attempt to assist and support, and we want to do that not just for product-specific cases in advice, but also let's find a way and topics for CPATH to look into internationally. So there are other things perhaps, but I stop here just to support the idea of our interest looking into that together.
Thanks, Ralf. That's good. Smithy, you've now heard different perspectives, and if I ask you first to introduce yourself, of course, but then in light of that, we probably need several tools to address different of the issues we're talking about. But are there some areas that you immediately think that, wow, that sounds attractive for something like CPATH2?
Absolutely. Thank you, Tomas. Thank you to the rest of the panel. This has been a wonderful conversation to be a part of. I'm Smithy Heavner. I'm CPATH Senior Scientific Director for Real World Evidence in the Data Collaboration Center. Been with CPATH for 4 years, and in a previous role, I led the Cure Drug Repurposing Collaboratory, which was a partnership with the Office of Medical Policy, specifically tasked with looking at how can we leverage the electronic health record to study how drugs are used off-label and support label expansions. And so I'm going to use that as an example of some of our work there. I think that what we're talking about is very feasible. A series of tools, resources, a toolkit, probably a set of toolkits. I might think of a toolbox. Actually, is probably getting close to a whole garage at this point, though. But there are some areas that are certainly lower hanging fruit, more feasible, or there's already practical experience to point to. I think that there are obvious challenges that have been brought up about the comparability of the groups, where we can all pretty easily think of an example where a clinical trial for a specific drug is dependent on a genetic diagnosis or specific outcome measures that we would not see in real-world data. We might see them in registries, but in the EHR, we're likely not going to see the kind of genetic testing that we would want for a lot of the diseases we've talked about this week. A lot of the outcome assessments are sometimes used, but there's also a question of how well that's actually being documented in clinical practice. The example I frequently will give is, if you ask someone, if you want to use smoking as a variable in your analysis, you have to recognize that in clinical practice, and speaking as my experience as a nurse, we don't always ask the question in the right way. You're supposed to say how often you smoke, a very permissive way of asking it, but instead what happens is the nurse walks in and says, "You don't smoke, do you?" Which is going to get a response. It's a very biased response to that question. Similar things happen with the PHQ-9, the GAIQ-7, any of the mobility scores that we would see for neurodegenerative diseases. There's going to be a very practice variation in how that data is collected. There's also specific things that inform the way data is collected in the clinical practice, that some of which we've talked about this week. I think specifically about Dr. Tanner's comment about the payer informing which drugs are used. The payer also informs what tests are done. There's also heterogeneity in practice. And one of the examples from CDRC, I promised I would get back to it. We were specifically looking at COVID-19. There were some obvious challenges with that data and that use case, the rapid evolution, the progression over time, the introduction of vaccines, new treatments, and the way that we approached gathering data introduced a lot of limitations. We had to de-identify, so we didn't have dates. We couldn't actually document and understand the relationship with individual patients to when vaccines were available. So we could know patients were vaccinated, but we didn't know which patients would have been eligible for vaccines even. But in COVID, we found something even more sinister, and this is a very extreme example that I've used before in our internal conversations in CPATH. and we're working on some papers around it. But one of the clinical indicators, the clinical biomarker that you look at in COVID-19 is something called a D-dimer. It's a measure of clotting activity in the body. And that microclotting is a really important factor when you think about patients that are severely ill. It's also a test for something called a pulmonary embolism, which is a very severe clinical outcome that can happen. And if you test a patient for that and they test positive, there's lots of additional information you need to gather. There's specific tests you need to do to assess what's going on. And so in talking to clinicians, because we noticed there was some strange distribution in the availability of that variable, we realized that not only are they basing the decision to conduct that test on whether it's clinically relevant, they're also thinking about their medical liability. And so you only see that in very specific cases, the most severe patients. And so if that's part of the model that we're constructing, we're already changing the sample that we get. Whereas in an RCT, you have a protocol that guides what tests are done, when, how is that data collected, and we have to make sure that the study that we're designing is going to have the variables available in our real-world data source. And this might change depending on whether it's the registry again or the EHR. So there are lessons learned, I think, is a great place to start talking about some of these questions to get at how do you evaluate your research study, the data source that you have, what are the potential sources of bias that might be known or unknown, how do you engage with stakeholders, whether those are patients, clinicians, regulators, to ask targeted questions to understand the fit of your approach. And then getting into more advanced questions, some of the modeling questions that we've discussed, some of the outcome measures, that how can we approximate those in an externally controlled arm. It's probably a much higher bar and much longer conversation that we need to have.
And yes, and I understand that this can be an interesting, but perhaps not everyone sort of dares to go into this exercise, but I think it's quite, well, it's essential. And the reason why I say dare, Smithy, is that perhaps just taking randomized controlled trials and creating external control groups and compare the control group to the placebo outcome in that would be very informative.
Yes.
Yes. Okay.
Absolutely.
Questions, quick questions, perhaps quick answers. Rob, I'll start with you because you said this is becoming, it's most difficult when you have an option to, this is how I heard you, when you have an option to do a randomized control, but you want to do it as an external control. That's most difficult from one perspective. But on the other hand, it's equally most difficult when you actually don't have an option than anything than an external control group, because then you really need to make sure that we're looking at the drug effect when you don't have anything. And because if we interpret that wrongly, patients may not get access to this new drug. So would you agree with that perspective that they're actually two different things here we're talking about? If you have a choice, you have a choice, and that's good in one way. When you don't have a choice, then it's even more important that we actually can interpret the outcome of the external data.
I'm not convinced that was a quick question.
No, that was not a quick question. So I'll ask you the quick question and you get the, yeah, that was a good one. Yes, you agree.
Two different complexities.
Yes.
Where we are forced not to do an adequately powered RCT because of limited patient numbers, for example, there is the challenge of paucity of data and the consequent challenges of establishing that a true drug effect exists and trying to quantify its magnitude with sufficient precision. The greater challenge to which I was referring, and thanks for the opportunity to clarify, was that if a company is trying to propose use of an external control arm where an adequately powered RCT is feasible, the mindset that I often see in the interactions between the company and the regulator is the regulator says no until you can convince me that your evidence will be as good as that from the RCT. And that's impossible. Practically impossible. And I like what you said, Marie, that actually the standard needs to be, will your proposal give me adequate evidence to establish substantial evidence of efficacy and characterize safety and make my benefit risk decision. But again, I'm not trying to be so critical because that's extremely difficult, right?
That's a bar that has to, like, the regulations don't change, right? And I think, like, if the sponsor can do an RCT, the FDA is going to say you need to do the RCT, right? That's what happens, right? You know, it's not like, oh, well, if you want to consider this, it's usually only if the RCT cannot be done that the randomized, I mean,
and
that is not always, maybe there are exceptions to that, you know, but it would be unusual.
And I often use the same guide for companies that you espoused, which was baked into even ICHE 10 back in the last century. This is going to work where your effect size is bigger than the plausible magnitude of bias that you might be introducing in the way you generate that effect size. But it's hard for everybody to quantify what that magnitude of bias might be. And I think that's really where people could benefit from some additional research and some additional support. And I think it links, Angela, to what you were saying. We need to have an idea as a company whether we're even on the right track here before we invest the resources to go and have conversations with regulators.
Staying with Angela and you, Rob, I'm sure there are people out there saying, but this is a recognized problem. We're handling it through sophisticated statistical methods, propensity score matching. We do whatever sorts of tests to look at potential unmeasured confounders, etc. Can you, in a few words, summarize why you still see a need to move beyond that for this conversation within sponsors team, but also between regulators and sponsors?
Well, what I would say is that if you're trying to handle it through the analytic process, it's too late. It actually involves a lot of upfront consideration and evaluation about fit for purpose, data quality, and so forth to really understand the feasibility of the approach. And that's where I think, you know, when you move ahead with a strategy without having done that planning, you introduce a lot of uncertainty and anxiety and potential for, you know, late stage rejection by a regulator. So, I mean, my argument is that's far too late if you're and really infeasible if you're relying on analytic approaches. So, yes, we have sophisticated analytic approaches, but they can't always solve for all the problems that exist in the real world data sources.
Want to comment?
I just add one word that, I mean, there's many different types of sponsors. We've got sort of large pharma representation that have all the skill sets in-house, but there's many, many smaller companies trying to do important research who just, they may have an in-house statistician, but honestly, your regular statistician doesn't get trained for this. And so it's someone who's sort of partially educated, but could really use more than will ever be available and ever be possible to publish in a regulatory guidance, because again, you're not trying to write textbooks on how to do this, right? And so there's a gap in competence or capacity.
I agree, and I think that, like, you know, maybe that could be a focus for this framework or tool, like some sort of training initiative could be really helpful.
Sorry, Ralf, go ahead before I go back to
So we need content for the training, and that is part of it. But I think the toolkit could also be a toolkit to play with. So if you think of the simulator that Klaus has shown us, which is hugely important, it is an instrument for exploring parameters and changes in selection, etc., and patient characteristics, treatment effect sizes, etc. So I think if we scope, we have data quality matters. We certainly want more and better tools even for handling data quality and for measuring data quality in relation to research questions, etc. I think we have principal questions. You raised the question of feasibility. We leave that aside because that's sort of not the difficult question. The other question then is, if there are choices, so to say, is not also a single arm trial a choice? Yeah, that also needs to be considered. We have given this a lot of thought and thinking. And the last comment then is we need to find out the principal aspects that, as I said, abstract from these matters. What is the type of um, uh, um, methodological, scientific, clinical discussion we need to have that goes beyond the mere question of data quality and feasibility, etc. And it perhaps even has a element where the regulators would currently say this is the point estimate, and we have, I think, to the best of my knowledge, not ever accepted that we are regulating based on having shifted the point estimate by some form of understanding of the bias that could be part of it. And so I think that there are some of these elements, and again, we would be happy to be part of such a conversation where we look into that broadly enough to then have some suggestions for actionable points. Yeah.
I'll take a couple of questions from the floor. I saw your pen moving up and down, Rob, but I'll also take a question over from there. I see you as well, Cecile.
It's still morning, so good morning. Thank you for this really insightful discussion. My name is Tina Morrison. I'm a former FDA, 17 years focused on advanced computing methods for medical products. Around this conversation and connecting some dots, that's the theme of this meeting from the conversation this morning about, the value of the patient, the value of therapies for patients and their families, and thinking about the regulatory bodies saying if an RCT can be done, well, let's do that. I wonder what the pharma companies are thinking about in terms of the cost benefit analysis. And by cost, I don't just mean financial. So maybe it could be the value benefit analysis of if there's data that exists and methods that can be developed to use data that's already been collected to answer important questions. Does that lead to a more reasonably priced therapy for patients rather than the time and extensiveness and complexities around an RCT to bring a therapy to patients? Are folks thinking about this kind of conversation and thinking about the needs of therapies using real-world data as compared to traditional clinical trial methods? This is not a space I'm familiar with, but feels like there's a piece of this conversation that isn't just about, well, if an RCT can be done, let's do that. Because yes, we do want rigorous science, but there's a balance of the costs that then get translated into the therapies and what is the value of the return on investment for harnessing real world data in these creative ways. I welcome any comments on a discussion like that. Thank you.
Yes.
Yeah. So this is something we've had some early conversations about at CPAP. One of my colleagues in quantitative medicine, Grace Lee, who some of you all have heard from this week, she and I have been talking about what would, for example, a return on investment calculator look like if we think about real world data. It's a big challenge to try to measure, but in early conversations. But that is one way that we could think about the financial incentives for real world data. As far as whether that might lead to changes in the cost of therapy, that's probably out of our scope at CPAP. But I don't know if anyone else is willing to comment on it.
I just want to touch on something you mentioned, and Kay mentioned this morning as well. We're all trying to understand the clinical or patient-relevant value of our drugs, right? And that's not relevant only to regulators. Regulators, you need to pass that magic benefit-risk bar. But I think it's been mentioned, it's so important to patients, but also to HTA payers. Because in the end, they're going to look at the potential clinical value and see if they're prepared to reimburse it, for example, in Europe. So what we're talking about, trying to understand the clinical value in a situation where we can't use more precise tools, if I'm allowed to use that, I think is so important, because we are, again, developing very promising drugs influencing long-term disease progression, etc. And how do we understand the value? We had the Alzheimer's discussion yesterday where Michael talked about early interventions, and that was the theme for other diseases as well. We can't do randomized control in that setting, to my mind. Cecile.
Yes, just along this line, I think that, so thank you to all the panelists for this very good conversation. And I think that going back to where my heart is in pediatrics, even if you can run RCT, I don't think it's ethical if there are alternative solutions that can be leveraged to minimize the burden on patients and their families, as we have seen, like it throws their life out of the windows, all the logistics of trials. And I think that regulators here have a role to incentivize companies to really look at generating the right level of evidence in real-world settings and minimizing that burden. So maybe more a comment than a question, but
No,
I
but
think
it's fine.
important.
And another remark. I got so mixed feelings when I see all the people in front of the microphones because I'm so happy that it's engaged people. I also see the watch down there. So we're going to take a couple of more questions. But Rob, your pen went up earlier, so maybe
Take the question.
What? Take a question. Okay, so I go that direction then.
Hi, it's Suki Malhai. I head up the DMD program at Wave Life Sciences. So I want to piggyback on what Cecile has just said. And my question is, how are we going to inject a level of urgency into this discussion and getting somewhere on this? Because, and a lot of what, Rob, you've been saying resonates with me. Here we are in the process of designing a phase 3 study for our program. And we are in a place where we'll have many patients that will now need to be on placebo for up to 18 months. And during that time, some of those patients will fall off the cliff in terms of ambulation. So I think ethically, I would like to talk about where is the urgency in supporting these patients, these families, so that there's a dearth of evidence that's already been generated, but we're still, doing the same thing. So that's my ask, and I'd be happy to be part of any conversation, any framework, any group that is going to be looking into this, just because we owe it to the patients that we're working with.
Yeah, I think we owe it to the patient that we generate conclusive evidence as fast as possible, right? And no one is served by delays. No one is served about generating the wrong answer.
No, but I guess my point is, the use of real world evidence, whether it's to augment placebo arms or just using real world evidence and not having a placebo arm at all. How do we generate this framework to allow for us to do that? Because we understand, and I'm not going to take too much time, I understand the regulators' need for all of this data, but we have a lot of data as well. So, how are we going to bring all of that together so that we don't have to have so many patients on placebo for such long times?
True. And I'm sure you watched Rob's slide with the design where he did have an initial placebo control and an external control running in parallel, matching the external control. He didn't say it, but that's what he had on the slide. Matching the external control during that first placebo period, maybe of a year in the Duchenne's disease. So you can then continue into non-ambulatory patients, which is not a new idea. It's been done, but it's not done often enough in my mind.
Can I just interject
Oh, yes, sorry.
I think the frame I understood is about, we're not here to discuss much about the real controlled trials as such, because we wanted to look into the opportunities here. But I do want to say that I think when I think of randomized controlled trials, these are a priori those clinical trials, or these need to be clinical trials that are scientifically well designed and ethical. So that there are challenges, of course, but let's not contrast with something that wouldn't be sufficiently ethical or scientific. This is something I really want to distinguish. This is also our role as regulators. Whatever the type of trial is, we want to make sure it's scientifically sound. That is the basis for also being ethically acceptable.
Sometimes there can be disagreements about what's ethical and what's not as well. I've encountered this. There can be, between a company and a regulator. So
yeah, and this is an important point. We need to think of utilities. We need to think of, you mentioned the public health value, financial values, etc. There are frameworks to bring this together, either quantitatively or qualitatively. And I think surprisingly, one of our recent guidelines on the single arm trials calls for better qualitative reasoning. And I think that is a particular area where not just the data and models, but really argumentation and qualification and balancing is something we can work on. Regulated science research involves social sciences. And I think that is an important aspect where we need to find a way to build that framework of thinking. Yeah. Just thank you for
that's great. I'll continue until someone walks up this. Yes. Okay, good.
Okay, I'm Jillian from the Galactosemia Foundation. My daughter was in a trial with placebo, 18 months, got the phone call, it was awful. I also am an ICU nurse. I come from a background where I understand placebo. I look into it. I went to a really awesome conference and talked about even the placebo aspect of signing consent. Like, it's there. This is another problem with ultra rare. You get 40 patients in a room, 10 people are on placebo, and they're very active moms, and they talk and they start comparing the serum that they're given and they unblind it. How is that addressed?
So I don't want to go there.
Easy question.
And
Floated, but to see that from my, like, I've
No.
had mom and the scientific rigor of a study. Like, I want the rigor, I want the science there.
Absolutely. And the reason why, because I think that is such an important issue, functional unblinding, because that's, I understand what you're talking about, is such an important aspect. And that's why I stressed blinding is so essential to interpret the randomized controlled trials. But I think it is a little bit not, it's not the topic of this session. And Klaus, next year, we'll have a session on.
I have thoughts.
What?
I have thoughts for next year.
Yeah, good. Thank you.
We'll talk after.
Yeah, because it's, we spend easily an hour, and I'm very happy to be part of that because that's so important. Yes, please.
Penny Dax, AFTD. And a toolkit for external comparator arms could be one of the most profound tools that could come out to accelerate drug development that I have seen proposed in a while. I would say that in hearing what you guys are describing, I'm not hearing any voices about the academic and the nonprofit that are usually the sources. So right now we have multiple, I think it's 6 different companies all doing phase 2 or phase 3 drug development for a single genetic arm of our disease. Every single company is going to 2 specific natural history studies to be the entirety of their confirmatory evidence, external comparator. And every conversation that we have is, well, are they regulatory grade? I don't know.
Yeah.
But those academic scientists can't go through the in-depth guidance from the FDA of what is real-world evidence because it's not their headspace, it's not where they think, it's not their language. We need a toolkit that can truly be a guide for what the academic scientists and the nonprofits, I mean, Farah over there, right? They were the ones that founded and led the natural history study in their arm. We want to be partners. But when we hear what is regulatory grade and all of these really complex terms, we need a toolkit that is going to meet the needs of our audiences so that we can then be partners in bringing those tools forward and attract drug development.
Yeah.
And I would also consider having a consultants that we could then tag, that we can pay for. We'd love to pay for a consultant that CPATH and the regulators signed on to say, hey, work over the course of 6 months or a year with the natural history study leaders in our space to help them understand what they could do and what the budgets would be to get to what the regulators and the companies need. Thank you.
No, but thank you for that point, those points. And yeah, I agree that sometimes the internal conversation, that was why I referred to that within the company, is so important for this as well. And regulatory units are usually quite conservative. I agree with that.
Tomas, just
Yes.
for not-for-profits, for academic researchers, we also offer scientific advice, and you don't need to budget for it. I think
That's true. And
really important.
early
These
interaction
type of questions are coming to us, and we're happy to look into that, even as a question related to a registry. So it's not uncommon. We can talk later, but we welcome this, we invite it, and we have published this the other day.
So I want to hear the 2 questions, and then we'll see how much, 2 last questions, and then we'll see how much time we spend on answering if we do it in the break. But can we just hear them, because I want to hear what both of you are saying.
Yes, so Rebecca Fuller, CHDI Foundation and Movement Disorder Society Electronic Clinical Outcome Assessment Committee. As we're in Movement Disorder Society, as we're making electronic versions of our assessments, that is going to sort of unify administration of the scales and potentially on the back end, data collection. And we could potentially also add training to make these available to those in clinical practice so that it can be real world evidence. We need support to do that, but it's something that I've been thinking about. And then all those assessments could potentially be a, good arm for real world assessment, real world, data.
I think
I also have thoughts.
that's also, I think that many of us have, because I think it's a really good comment how we can link between what comes out of clinical practice and trials. Peter, last words of wisdom or question or?
It's really a question, and I want to focus on process, not on substance. In 1997, Congress enacted the FDA Modernization Act and included an entirely revolutionary procedure whereby the outside world, whether academic or commercial, could pose a question about a proposed protocol, and FDA, this is called the Special Protocol Assessment Process. FDA is required to answer, and not just answer, but to say not only yes or no, but to add on why if it's a no, give the rationale. And on top of that, they are required by law to say how it could be improved so they would get a yes the next time. Now that sets up a dialogue, obviously. It's not just two, one question and one answer, because if the answer is no, the company or the academic can go back with a modification until there is a resolution. Moreover, I think, Marie, probably in your experience, usually this is done very informally rather than very formally. But if it can't be done informally with, say, a division that doesn't like the special protocol assessment, you can force it. And with a process like that, you can get to an answer. And the final thing is, under the user fee provisions, there's a limited time period for FDA's response. Marie, is that a useful process?
I mean,
Is it already in place?
Yeah, I mean, as I mentioned, FDA are very open to discussing early and often with a sponsor, any concerns. I would say that in some situations, it's a no because it's the data isn't fit for use, or there are major study design concerns around comparability. So the sponsor goes back and can think about other options, but so I wouldn't say that it's always a success, but I would say that yes, we encourage people to come early and often. I won't comment on that program specifically, but and we have a real world evidence mailbox where we can pass queries on to the relevant division. We have our advancing real world evidence program, where we encourage people to submit early proposals for sort of dedicated feedback outside of the traditional channel. So there are options, certainly.
Thank you. And I'm sure we could have gone on for quite some time, but I'd like to stop here, and I'd like to thank the panel for reflections and answering questions. I'd like to thank you for asking the questions and saying